I'm Katie.
And I'm Vinny. And this is Learn Real Good.
Real science.
Good times.
Nailed it.
That's our new tagline. And we're trying it out here.
Yeah.
I like it. Real science, good times. That's a good tagline.
I hope they're all true. I mean, we definitely the real science. I hope good times. Can we put an asterisk? No guarantees.
That's really a lack of confidence.
I know.
I'm having a good time already.
I know. We're very easy to please, you know?
Why not? Why shouldn't we be?
Well, we're talking about science and we're being silly about it, which is the name of this podcast and our thing.
That's a good time guaranteed right there.
Okay, so we're officially removing the asterisk.
Yeah, I don't think we need
good times.
Yeah, I feel like a tagline of real science, good times, / good times not guaranteed. It does not feel like a great slogan.
What about good times ?
No.
Oh, this is
I'm
really...
having a good time.
Okay.
Legit good time. Why do you need to qualify it?
We're learning that I have no confidence in my ability to have a pleasant time with myself.
Are you having a good time now?
I am.
Okay.
I'm relaxing into it.
There you go.
So for our new listeners, this is a comedy science podcast where we share some facts at the top of the show, and then we have a wonderful guest.
Yeah, a Canadian grad or undergrad student.
Studying in Canada.
You
don't necessarily
Correct.
know.
We don't know.
Canadian.
We don't check passports.
No. Or COVID passports. It's all fine.
Yeah, because it's all remote.
Yes, exactly. Yeah. Of course.
So just as long as you claim to go to a Canadian university.
Anyone can be on this show.
Anyone can be on this show.
Yeah.
We're going to question you.
Yes.
Yeah. First, can you count to 10?
Yes, that's we ask
one.
Every guest that.
To count to 10.
It's not a sound check.
No, it's a math check.
Yes.
It's not really math counting.
Yeah, it's just counting.
Yeah, that's not an operation.
Can we cut that?
What? Why would you cut
We don't
that?
know what is counting, what isn't counting.
Is counting an operation? Is counting math?
Whoa.
Our numbers, just the existence of numbers, the concept of numbers, is that math?
We need mathematicians on this show.
Someone's got a definition. This is more like the philosophy of science kind of thing.
Whoa. Okay.
Yeah.
Yeah. This is getting above
this is
my
above
pay
our
grade.
Yeah. This is way above what we normally talk about.
I mean, I'm an ecologist by trade. That's my thing.
But you did a minor in philosophy.
Don't bring that up. It's a minor.
Why are you ashamed of it? Don't bring that up.
It's not even on my degree. I don't think it didn't make the grade.
Okay.
Yeah. So I don't know. I feel like I have a shuddering lack of confidence. That's what this whole intro is unveiling.
Where's that class? Do a minor in that.
Confidence?
Yeah.
That would have been great. Should have done that. Shoulda, woulda, coulda.
Shoulda, woulda,
coulda.
coulda.
That ship has sailed. Well, this has been good times so far. But
yeah, we haven't gotten to the real science yet.
I know. I'd like to.
Okay.
So it's time to share facts. So me and Vinny come to each show with a hot science fact that we're going to share with each other.
Correct.
Vinny cuts to the chase, and I like to vamp up to the fact for about 20 minutes and then just spit out the fact.
So 10 minutes into the vamping.
Who goes first?
Go for it.
Oh, my fact?
Yeah.
I don't know why I'm so surprised that I'm going first.
It's your lack of confidence.
It is. I mean, that's
This show is all about getting you to believe in yourself.
You know what's amazing is I've had this window open all day. Oh, here it is. I have a different thing. Okay, great. Are you ready for this?
I'm psyched.
Okay, so this comes from Science magazine, very recently published hot new science information about how our human cells how our cells fight bacteria.
You sounded like an alien there.
I know.
As a fellow human, I have cells.
I also have human cells.
Yeah.
I think the reason why is because I'm going to be talking about humans fighting bacterial infections.
Sure.
But you could argue, once a bacteria has entered your body, is it now one of your cells?
We're into the philosophy of science again.
Right. I only have a minor, not a major.
Our number's math. Are bacteria in your body human? We don't know these answers.
Well, they're not human cells, but they're our cells.
Are we not
person
human? If you prick us, do we not believe?
Okay, let's stop quoting. Is that Shakespeare or Seinfeld you're quoting? Okay. Okay, so how we've learned a new way that our cells fight off infection. So you're aware that we have cells whose job it is to be part of our immune system and go around and fight infection.
Yes.
Right? And if they notice there's a bad guy, right, the antibodies, if you have them, will, flag it for destruction. You have these cells floating around that'll destroy the bacteria.
Only you could see Katie's arm gestures.
It should be a video podcast. A lot of hand gestures.
A lot of robotic arm movement, karate chops.
So that sounds familiar, right?
Yeah.
Okay. Well, our non-immune system cells can also fight bacteria. So if a bacteria invades, say, a skin cell, it's not as though that skin cell's screwed now and is going to automatically succumb. It can fight it itself inside the cell.
The cell itself.
Yeah, non-immune cells can fight. And we knew this.
Okay.
We didn't know is necessarily how they were able to do this. Now, this is the new fact that was discovered.
New goss.
The material that they use to fight bacteria intracellularly, inside of a cell. So, going back to our immune system cells, they can sort of send out these chemical signals called cytokines.
I love these hand
Pew,
gestures.
pew,
Yeah,
pew.
your little like antennas coming out of your hands.
And they just tell like non-immune system cells, you guys make this chemical. And this chemical is basically soap.
Oh.
So it's this protein.
Phospholipids?
No, but I love that your mind went there. So we will come back to that. So how does a soap work, Vinny? Do you know?
Yeah, it's 2 parts, one that is hydrophobic and one that is hydrophilic, or one oleophilic and hydrophilic. It's like one part that attracts grease and one part that attracts water, and it acts as a bonding agent between oils and waters.
Yeah, it's hydrophobic and hydrophilic. Yeah, so something that likes water, something that doesn't like water.
Yeah, oleophilic being grease attracting.
Yes, likes lipid, likes non-polary things,
Yeah.
like lipids. Yes. So it sort of mediates the two types of fluids that don't get along well together in order to, if you wanted to wash dirt and grease off your body.
As I
soap,
often do.
I'd like you to do more. Soap will sort of allow it to be washed away by the water in the shower. Okay. So our cells can make a detergent, a protein that has hydrophilic and hydrophobic components, and what that does is it destroys the bacterial cell membrane and can help fight infections. Now here's the final cool bit. Why doesn't it destroy our own cell's membrane in the process?
Trick question. Cells don't have membranes.
Great try. They do.
Oh.
It's because our cell membranes, being animals, have cholesterol, and cholesterol blocks it from having an effect.
So it's like a detergent repellent.
A detergent deterrent.
Oh, a detergent deterrent is much better. That's good.
I can't believe I got that before you. Yeah. Cool, right?
That is cool. So
Yeah.
basically, when we're attacked, our body says, hey, make soap.
Yeah. There you go.
And we're immune, or the cell membranes aren't going to be destroyed by the soap, but the
Correct.
bacteria membranes are.
Correct. And what's cool is it's not even like wimpy bacteria. Even like the super hard to kill bacteria can be destroyed by this process.
Soap.
Yeah.
Human soap.
Human soap.
Wow. I love it. Good fact.
No, thank you.
Nice.
I didn't make it. I don't know. I took credit for it there.
You found it.
Should I have confidence about this fact?
Yeah.
Okay.
Yeah. You had to select it.
Thank you.
It
Thank
was
you.
a good choice. You have
Yeah.
great taste in science facts.
Oh, thank you. And that comes from Science Magazine.
Nice.
Yeah. Well, it's your time.
Yeah, my time
Your
to
turn.
drop some
shine.
science.
Yes.
We'll see if it shines.
Okay, let's see if you can beat deterrent.
Deterrent, I can't already.
Okay. Wow. Bowing out. Okay,
Lack
let's
of
hear it.
confidence as well.
Oh, wow. It's really sad.
Well, you had a really great one. So
Okay.
here we go.
Mm-hmm.
So birds, right away.
Okay, I'm thrilled.
Yeah.
Birds, yeah.
Birds.
Love
They
'em.
fly around. A lot of them migrate,
Yeah.
and they need to know where north and south is.
Mm-hmm.
And what's the leading theory on how they know where north and south is?
Magnets.
Correct.
Yeah.
Not just any old magnets. Not just like fridge magnets.
They each have a compass.
Yes, that's right. Each one is born with a little compass. No, the Earth's magnetic field. And it's always been a bit of a myth, like some theories of like, how do they know where the magnetic field is?
Yeah.
Well, recently they found these proteins called cryptochromes
Whoa.
in birds' eyes.
Okay.
And in the lab, cryptochromes are sensitive to magnetism. So this has not been proven, but just the fact that they found these magnetically sensitive proteins in bird eyes, they're thinking that birds may be able to see magnetic lines. It's a possibility. So nothing's, again, like I want to be pretty clear about how unconclusive this is. All they've done is found a magnetically sensitive protein in a bird's eye. So that does not automatically follow that they can do it, but it doesn't not mean that either.
Whoa.
So these cryptochrome, there's several magnetically sensitive proteins, and some of them were like theorized to exist, and then they like looked in a bird and like, holy smokes, we found this protein that matches all the characteristics of a magnetically sensitive protein.
Do you know how they were able to determine that is magnetically? Did they like isolate the protein from their
So what
eyeballs?
they did was simulate what some proteins characteristics of a magnetically sensitive protein would look like, and then like built a computer simulation and test and see like, is this magnetically sensitive? And it is. And then they looked in birds' eyes and found proteins that match the characteristics.
Wow.
And so birds' eyes have chemicals that are magnetically sensitive and may well be linked to their sense of magnetic direction.
I mean, that makes a lot of sense. Like if I try to think of what other way they could perceive.
I mean, something in those birds has to be magnetically sensitive.
Yeah. Okay, what else could it be if it's not the eyes?
Their compass first, possibly the little baby bird compass that they're born with.
Yeah.
But if they can't read that, maybe it's the proteins in their eyes. Mm-hmm. And if not that, then maybe they have a really great group discount on iPhones.
That seems likely.
That's my third theory.
Wow, fascinating. Yeah. Very cool. And so that's just all preliminary. And now I'm sure a bunch of
people are going to try to show
ornithologists will be diving into that.
As an eagle would.
Wow, that's very cool. That's like shocking.
Yeah, very surprising.
Birds are neat.
They fly. Already an amazing achievement on any animal level.
You immediately gain so much respect for any entity that has the capacity to fly.
I just tell gravity to take a hike. Good work, birds.
All right, well, that was a great fact, Vinny.
Thank you. That was yours.
Yes, well, I think we should both feel more confident about our fact finding and fact delivery.
I think those are the two greatest science facts of all time.
Well, I think we're in for a bunch of fun science facts. Segue
to our guest. Let's say hello to our guest. Today's guest is Isabel Hilgendag. She obtained her BSc in Environmental Science at Laurentian University in 2018. She's currently doing her MSc in Biology at the University of Waterloo. Her research investigates mercury in Arctic marine food webs. Isabel has also solo traveled over 15 countries, is fluent in 3 languages, and loves hiking and blogging in her spare time. Hello, Isabel.
Hello.
Welcome.
Welcome to the show.
Thanks for having me.
Oh, this is a delight. I can't wait. And we're going to use all 3 languages throughout the show.
We are? I don't even know what the languages are.
English.
Yes.
Let's guess. Right off the top.
English.
French.
And Swedish or Danish.
No.
German.
No. Yes. Good job,
German.
guys.
Nice.
I was trying to guess. Is that related to your familial history?
Yes, it is.
Okay. I was trying to guess the origin of your last name.
We were in that area. That whole...
Yeah, it gives it away a bit.
Nice.
Well, welcome. And this is going to be great for Katie because she also studied food webs.
Yeah, food web buddies.
Food web buddies?
No pressure, yeah.
Me and Isabel go to Food Web Con every year.
Oh, yeah.
Each other there.
Well, welcome, Isabel. And I guess first thing to ask you is mercury in Arctic marine food webs. Can you tell us a little bit about that?
Oh, yeah, there's so much to say. I know it seems a bit weird that there would be mercury in the Arctic. But yeah, mercury is important to study because, it's a heavy metal and it's known as being a neurotoxin and an endocrine disruptor. So that just basically means that it'll impair brain function and reproductive organs. So 2 very important things. So it's important to know that. But so mercury is emitted from all around the world, global sources. The 2 main human sources of mercury are from fossil fuel combustion and gold mining. But there's also natural sources as well, such as volcanic eruptions or geothermal activity. So basically what happens is mercury is emitted into the atmosphere or the air, and then it's transported around the world. And then eventually it'll deposit. And one of the places it deposits is in the Arctic. So, and once it's there, then it'll go into the food chain and be lead to really high concentrations in top predators.
So what is it? Is there anything about the Arctic that receives an above average amount of mercury? Does it have something to do with ocean circulation or anything? Or is that just a place you chose to study it?
Not necessarily. We have high concentrations of mercury here in lower latitudes in Canada as well. It's just the reason it's so important in the Arctic is the fact that a lot of those, there's just a lot of like, it's called biomagnification when the amount of mercury increases a lot in the food chain. So up there, your really top predators, which I don't study, but those are like polar bears and things like that. So it can get to much higher concentrations. And then also it's important because a lot of people that live up there, such as Inuit, they really rely on those top predators as food sources. So there's more of a human interest in studying mercury in the Arctic.
So biomagnification is like the accumulation as like, so like something small picks up the mercury and then a larger thing eats a bunch of small things and then gets more mercury and then so on up the food web.
Yeah, exactly. Yeah. Like,
Okay.
so yeah, like in what I study, like, for example, you'd have like zooplankton, which are really tiny animals in the water. And then eventually they'll be eaten by something that's bigger and bigger. And then, so fish will then have much higher concentrations.
And what is it about heavy metals that is so prone for bioaccumulation? Why can't we just get rid of them?
Well, what's special about mercury is that it has like, it can have different chemical states or different chemical forms. And that's what makes it a little different from the other heavy metals, is that it has a more toxic form. There's a form of mercury that's a little less toxic, but then there's also a form that's more toxic, and that really causes it to bioaccumulate.
And so how much mercury can I eat before it starts becoming a problem?
I don't know the number for humans because it's pretty variable, but I mean, for fish, like the threshold for like Canada's general guideline for like fish consumption is like 0.5 ppm, like kind of low, but like
That's
the thing
pretty
is, yeah, once you eat like lots of it, then it become a problem.
So I shouldn't eat too many thermometers.
Yeah, exactly.
Okay, got it.
I've told you it's a destructive habit.
I just want to know what the temperature is inside my body.
Well, actually, for like, people that live in other places, not just the Arctic, our main source of mercury exposure, other than thermometers, would be from eating fish like tuna, for example, because they're big predators. And like, I actually know someone who ate a lot of tuna because it's like cheap protein, and then ended up like getting like numbness in his hands, and like, yeah. So,
Really?
From the mercury?
yeah, that's crazy.
Well, I know famously Jeremy Piven from Entourage got
What,
mercury
really?
poisoning from eating too much sushi.
Really?
That's also coincidentally the dumbest fact I know.
I think that could have been a great science fact.
There's a lot of warning about consuming too much sushi with mercury poisoning. Is that just because that's how a lot of trendy people consume fish? Or is there something about raw fish and mercury content that's related? It's just any fish. It doesn't matter if it's cooked or not.
I'm not actually sure.
Yeah.
I imagine it's just because people eat so much
see-
sushi.
I can't see how cooking would remove the heavy
I know, but there's
metals.
always warnings
Yeah.
about sushi rather than, I guess because there's a lot of tuna sushi.
I guess there must be just like really like fish that are really top predators in sushi. That
Yeah.
and people like
Yeah.
eat a lot of it because it's so good. So
I'll go to an all-you-can-eat sushi. We go to
Yeah,
town.
right? So-
We'll demolish it.
Just tuna.
Yeah, we eat a whole ocean. Sorry, ocean.
Don't share that on the podcast.
yeah.
Oh, like we're the only ones who eat an ocean's worth of fish.
I'm a vegetarian. Okay. Very cool. So can you tell us about your food webs? What are some of your main characters? So you study zooplankton, but what are some other big characters that are part of the food webs?
Yeah, so actually there's two main things that I study. One of them is actually Arctic char, which are like a fish in the Arctic. And they're pretty cool because they have the most northerly distribution of all fish, and they're really important food sources for people like Inuit. And then I also look at mercury in different food webs. So I look at mercury, so biomagnification, or the increase of mercury in the pelagic food web, which is like it's like in the water column. And then I look at mercury in the benthic food web, which is like right on the seafloor. So I'm looking at kind of how the rate of accumulation in those two food webs, and then also looking at kind of the role of Arctic char in the food web, and kind of that all sort of relates to mercury.
And you mentioned polar bears earlier too, because I guess I imagine they eat the char.
Yeah. Like there's also like, seals and like whales and polar bears, like those other characters up there in the Arctic. But I'm mainly just looking at up until like fish.
Okay. Yeah.
You got to
Cool,
cut it
though.
off at some point.
Yeah, right. Yeah.
So many animals.
Food webs are insane. Like when you start getting into studying food webs and all the connections, it is a bit of it's very fun and challenging, but also a nightmare to try to tease apart all these connections to know who's really responsible for who.
Yeah.
Yeah.
It
Too
is
many, too
really
many things
tricky.
eating other things. That's what I've always said.
So let's dig into so how do you study a food web even? Like, how do you see, like, what is the sort of data that you're looking at, and how are you able to trace the path of mercury?
Yeah, so I mean, we go up and collect all the animals, and then what I mainly to look at a food web, I use stable isotopes, and basically they're pretty cool. Like they can tell you kind of what is the animal eating, like is it eating more that pelagic food web I was talking about, or from that benthic food web, and then what's their trophic level? So where are they in that food
Mhm.
web? And if you have a higher trophic level, then it means like everything below you like are potential food sources. And so
Okay.
yeah, it's really cool.
Like, how do the isotopes indicate that? I guess that's my question, is like, is there one isotope that's more prevalent in the benthic level and the other one at the pelagic level, if I've got those levels right?
Yeah, sort of. So it's like it's called fractionation. So basically through chemical reactions, so you have like heavier isotopes and lighter isotopes. And through chemical reactions, like either either the lighter isotope will be preferred over the heavier one. So that's why, like for example, if I'm looking at my differences in food webs, I'll see that the pelagic food web will have a much lighter isotope, so it'll actually have a much more negative signature. So for example, like -22, compared to if it's like benthic, it's going to have a much more enriched signature, so like a higher signature. So, but for my research, that's like around like -17. So it's like within, like you kind of see that sort of range.
And these are isotopes of mercury.
No, sorry, these are isotopes of carbon. So so I'm
Oh,
looking at isotopes of carbon and isotopes of nitrogen, and those tell me nitrogen tells you the trophic level, and then I'm looking at kind of mercury. So I'll measure the concentration of mercury and then align that up with like my trophic level for each of those food
oh,
webs.
I see. Okay.
Now, I might have to cut this. It's been like over a decade since I looked at stable isotopes, because I also had those in my master's. Actually, I was starting to look at it and then I abandoned it. That's why
You're like, isotope? No, thank you.
It is
more like
hard.
isonope.
I mean, Isabel, you're doing amazing, but I was always like, I know these words and I still don't totally get it. So you use nitrogen for trophic level and use carbon for where in the water they are? Pelagic
Yeah, so
versus
like
benthic?
their food web, yeah.
Why is one good for one thing and one good for the other thing?
If you have a higher nitrogen level, like nitrogen value, let's just say like 9 compared to 12, that just tells you your trophic level, because it's what the like an organism that consumes higher levels of that value, it kind of just increases. It's kind of like mercury, sort of like
Right.
it's like increasing as you go up.
The
Where
concentrations of it.
yeah, yeah, kind of like that. And then whereas for carbon, it's based on the plants, like your base of the food web. So for pelagic, it's more like phytoplankton and stuff like that. So they just have a different way that they're incorporating carbon into their like bodies, I guess you can say, compared to like kelp or algae, which is part of the benthic food web.
Amazing.
It's very neat.
Yeah.
For me, like when I look at like the hot topics of conferences I went to, like stable isotopes was like everyone's talk had stable isotopes in the early 2000s, and then it became like CRISPR later on. That's about when I stopped going to conferences. But it was like everyone's using stable isotopes, and I still don't get it. But you're doing great. It's not you.
Yeah, I get it.
It's so complicated.
Yeah, I feel like, I mean, it makes sense. So you look at these chemical, like these elements and their isotopes to identify like where they are, what they're eating, who's who's dating who, I don't know. And then you're also looking at the mercury levels in, I'm presuming, in these animals. And what are you finding? Like what kind of correlations are you starting to see?
Yeah, so far what I'm seeing is that mercury is accumulating at a higher rate in the pelagic food web compared to that benthic food web. So that's
And
pretty cool.
why?
That's the part I'm still looking into. But
Yeah, I figured that might be a tough question.
Yeah, but like the pelagic food web, it's I think it just has a much more direct transfer, because like compared to the benthic food web, there's just a lot going on in the benthic food web. It's pretty complex. Like there's a lot of different kinds of animals down there, and they're all kind of along that sediment. And the sediment is also like a source of mercury, because like it contains bacteria that actually change the mercury into the more toxic form. So they're kind of like, it's kind of all over the place. It's not like a direct kind of linear, like, oh yeah, you're higher up, you have more. It's kind of just, it's sort of more all over the place. Whereas the pelagic, it's like more of a direct kind of pathway.
They're just eating the mercury straight up.
Yeah.
So you mentioned there's bacteria that turn the good, not so bad mercury into bad mercury. Do we know
The good, healthy mercury.
the chocolate mercury? Do we know of any microbes that do it the other way?
Oh
That
yeah,
go
like yeah. So the ones I was talking about are like sulfate reducing bacteria, but I'm sure there's ones that go the other way. I just don't know the names of them right now.
Sure.
No, I'm
Wow.
just curious. I wonder if people are studying like using the bacteria that do the good thing to get rid
Can
of.
we feed the fish eating the mercury the bacteria that switch the mercury?
Oh,
I'm sure there's
interesting.
people studying it, but yeah, I'm not. I don't know.
If they aren't, I will.
Yeah, Vinny will be
Yeah.
your supervisor. He just needs a healthy supply of thermometers to keep
I
him
already have
happy.
that. I've
Oh,
been
you
eating
have
them for
it.
years.
Okay. Okay.
Yeah, I'm really interested in reversing mercury poisoning right now.
You have a very strong
very pressing need.
personal investment
Yeah.
in that. So how did you come to this topic? Was there something that during your bachelor's or in your life that inspired you to study food webs or to study mercury, anything like that?
I mean, I grew up on a farm. So as a kid, I was like always outside, always exploring nature. And so, yeah, I guess I kind of got my interest, I mean, for biology that way. But then I guess it wasn't until high school when I took chemistry classes that I got really interested in contaminants and pollutants in the environment. And then it kind of just went, I got more interested in it when I took then my my bachelor in environmental science, where I was like, oh, this is really cool. I love this. And then, yeah, like, I guess like I mean, I did an undergraduate thesis, and my supervisor for that actually put me in contact with my current supervisor. And that's sort of how I ended up at Waterloo doing this project. But it's actually pretty cool. Like, I just, by following kind of what I've been interested in and taking those opportunities, I kind of just ended up doing something that I actually like really love and like always had hoped I'd end up doing, but like never really knew how to get there.
And like as a kid, like what was it like growing up on a farm? Like what was that connection with biology, the world around you? Like was there something on your everyday experience that kind of spoke to you, or just on a specific event?
I think I can't think of any specific events, but I mean, I would always be like collecting pond water and like looking at like the different larvae stuff that's in there, or like collecting toads. Like my mom would always get mad because I'd be like collecting like birds' eggs and like trying to warm them up to like have the birds hatch, or like I'd collect like I'd have like these jars of like water and like all kinds of like plants in them, and they just get like grow algae. I don't know. I was just always like just super interested.
All sorts of gross shit that Isabel's bringing home.
Yeah.
Your mom every day is like, oh God, what you gonna pull out of the backpack today?
Yeah, literally.
Yeah, just toads and jars of pond water. I love it.
I tortured ants, so you all have a dark past. All
I
biologists.
don't know if all of us. Oh, biologists,
yeah,
biologists,
you
maybe.
know.
Yeah.
Just curiosity.
Yeah, it's scary. You're curious
Do
about
some
the
dark
world around you.
things. I was also always fascinated by, we had a really big patio door at my house growing up, and birds would always fly into it and break their necks, and I used to find them fascinating. These
Wow.
dead birds. Okay, that's dark. Let's move on. Oh, I wanted to ask, so you study the Arctic. Where do you go to do field work? So you must, I guess you go to the Arctic, but like where specifically? How long do you go up for?
Yeah, so I do a lot of research around Iqaluit, which is the capital city of Nunavut. And so my field work, I spend days on the, like every day we go out in a boat and boat around Frobisher Bay and collect our samples. And when I went up in 2019 for 5 weeks. So that was pretty cool. Cause like for this kind of research, it's like, it's winter, like summer is basically only like a month and a bit, like 'cause there was just ice on the water the whole time until like right around like end of July is when I went up there. And that's when, I mean, there was still huge chunks of ice in the water, but you could actually like get some samples and yeah.
And now Katie and I are obsessed with the Northern Lights. So I got to ask, did you see the Northern Lights?
Yes, I did. And it was amazing. Everyone should see that.
I'm so jealous.
I know.
Never
equally
seen
jealous.
them.
Never seen them.
I've never been to the Arctic, but I've been to some pretty north places where people are like, oh yeah, we see the Northern Lights, and I've just never been lucky enough.
I've never left the island of Montreal.
It's not true. It's not true.
No, it's not.
Do not lie. Wow. So what is it like doing research in the Arctic?
Yeah.
Did you have to learn? I know a lot of people who've done research in the Arctic have to learn how to use a gun in case there's a polar bear attack. Was that you, Isabel?
Yeah, I had to get my gun license and yeah, make
Wow.
sure I know how to use a gun.
Is it because all the
terrifying.
polar bears are armed as well?
Oh yeah, no, they're feisty now.
Yeah, a little turf war going on.
Wow. Did you have like I know some, so based on what I've heard other people have to do, did you have like a personal bear bodyguard? So while you're doing your research, they are just sort of looking out for bears with a gun?
Not necessarily, because we were mostly on the water anyways, so it was pretty okay. But when I was doing research for something else, I was up in Northwest Territories, and that's when we had like, you know, some people with us from the community that had like no guns and were kind of our lookout.
That's scary. I don't want to go and face off against a polar bear. That's just my personal preference.
Yeah, pretty scary. Anywhere where I have to do a job and I need to be armed, that's a deal breaker for me.
No, you
Well,
wouldn't do it.
I couldn't do it. I don't have the confidence for a gun. We've established that.
Yeah. You need to have at least some level of confidence to be able to be armed.
So, Isabel, you mentioned your undergrad thesis. Can we hear a bit about that too?
Yeah.
Oh, sure. Yeah, that's going way back. It's not as interesting as what I'm doing now. So I was looking at differences between, so this was near Sudbury. I was looking at acidic and neutral lakes and kind of looking at whether different compositions of organic matter will change like the abundance of zooplankton. Because in acidic lakes, the zooplankton, there's much fewer of them. So we were trying to figure out, we did like, they're called in situ mesocosms, where we had like bins of different kinds of mixtures of organic matter. And we were trying to see, and we were like counting. It was a lot of counting of zooplankton. Like,
oh, wow.
Just a lot, yeah.
How do they move? Or like, how do you, it's like, feels like it would be counting a crowd at a concert.
Yeah, we had like these, well, like we didn't count them while they were like swimming around. That'd be like crazy. But we like collected them. There's like this, just like the way we set it up, we could like collect them. So like every day we collected those. And then later in the lab, we would, like put them in vials, and then like I'd go through and like count all the ones that are in the vials, and like also
goodness.
like identify them too, like what kind of zooplankton they are and stuff like that.
Give them names.
Yeah.
Do you have any favorite types of zooplankton?
Oh, I definitely did at the time. I thought for freshwater zooplankton, I think Cydidae are pretty cool. Like that's like the family name.
Katie's nodding.
They just
like,
look
that's my
really
favorite
cool.
too.
They look really creepy. Like zooplankton looks so creepy. It's weird.
They do. I had a haunting lab in my first year of undergrad where we had Daphnia, who would be my pick. They're so cute. I know it's hack to pick Daphnia, but they're so cute.
They're
They have
cute.
a giant eye spot.
They have an eye, don't have eyes, they have an eye spot. That's not good.
When you see them from the side, they're cute. When you see them head on, ooh, it's not so cute.
You said that about me.
True. If you could turn, that'd be great.
Yeah, I'll turn more.
So the experiment that still haunts me to this day is we were given this adorable Daphnia, which was the first time I'd see them. And you see it under the microscope, you're like, well, I am going to protect that with my life. And then you were given a mystery chemical that was either a depressant or a stimulant, and you had to watch because they're see-through, and you could see their like
Little heart.
Yeah.
The proto heart.
their little fakey heart, and you had to assess like whether you were given a stimulant or a depressant. It was very sad. Yeah. And they died, so it was
Wow.
it was a
Couldn't
dark
you just
day.
like play music for them? Like this is emo, and then this is some EDM.
Well, I don't know what their preference would be.
It's true. Maybe
Well, I
some
guess that's
EDM would depress them.
mystery. Yeah, I don't know. It might be a Daphnia to Daphnia preference. I don't know.
Yeah, maybe.
So you talked about counting Daphnia or zooplankton, like that's in the past. You're over that, but you're still studying zooplankton. So do you just have like an assistant who does that now, so you don't have to worry about it, or you're not looking at the number?
Yeah, I'm not looking at the number. Yeah, luckily I don't have to count them. Just when we collect them, so like when we go out on the boat, we'll like collect them with like a zooplankton net, which is basically like a really fine mesh net that we just like.
That's
pull
got to
along
be
the
very
water.
fine.
Yeah. Oh, it's like 500 microns. So
That's
really,
very
really
small.
Yeah. And basically with that, like once we collect them, we'll take out like anything that would eat them, like jellyfish or other kinds of fish. And then we bring them back to the lab and just sort through them, like just basically identifying them and then putting them in vials. But for this one, instead of counting, like we had to get like a quarter of a vial, which is kind of a lot if you're like, because they're so tiny. So yeah, it was a different kind of torture.
Right. Yeah. So much of grad work is torture. Research is torture.
In my experience, I always determined that the number one key characteristic to be an ecologist, a field ecologist at least, is your ability to withstand discomfort. That's
so true.
Right?
Yeah.
It is a rough biz.
Wow. Yeah, because you've told me also you like lived on an island for months at a time.
Not a tropical island.
No.
A cold, wet island.
A Gulf Island.
Yes.
Where it was raining 90% of the time. And your clothes got moldy.
I was moldy 90% of the time.
No,
Yeah.
not just your clothes. You yourself were moldy.
No, just my clothes, but it felt like they were part of me by then. Yeah, it was rough times.
And so, yeah, Isabel, have you is field research hard? Like, are there some wild stories from being in the field?
I don't know if I have any wild stories, but yeah, like it's just long days. and just like lack of sleep, which makes you go a little crazy, but it's a lot of fun and we get to go to cool places.
That's true.
Like I don't do I have any wild, I'm sure I do, I just can't think of them right
That's
now.
okay.
The cool thing though is like I got to, while I was up in Iqaluit, I actually got to help out with some other research. And so I got to like camp out in the Sylvia Grinnell Territorial Park. So that was pretty cool. It was just like me and someone else just camping out. It was beautiful, like, on the tundra. You could see like the fall
Wow.
colors of the tundra. It was gorgeous.
Unreal.
Jealous. We need to go to the tundra.
I'm there.
That's where the polar bears are. You gotta be careful.
Wait a minute. You have to carry the gun.
No. I'll hire someone, please.
Wait, well, so
Wait,
Isabel,
Isabel.
did you see a polar bear?
There was a reported spotting, but luckily it was just like I was false spotting. So, but we were worried because like we were like didn't know if it was like real or not. We're like, oh, like get the gun. I had like the gun under my bed. I was ready. Like,
Oh,
Wow.
was it just like 3 black rocks in the snow and someone thought it was like the eyes and the nose?
That's like the drawing of a polar bear in a snowstorm.
yeah, it must have. I think it was just someone must have had like a big husky or something and saw it from like, at the distance.
That's a
maybe.
large
I think that's what
husky.
happened. Yeah, yeah.
It was a perspective.
Yeah, it was a very close
Yeah.
husky.
There's a couple huskies, stacked on top of each other. No.
That's right. Yeah,
The trench coat?
they're doing a cheerleader pyramid.
Yeah.
yeah.
Wow.
Oh
And
boy.
so now here's a question: with climate change, is this affecting the how Mercury is getting into that food web?
Yeah, so that's like one of the important aspects of like what my research is for. Like with climate change, like Mercury is contingent on like its environment. So with climate change, our Canadian Arctic is warming at 3 times global rate already. So it's like it's rapidly changing the Arctic, and so this can either it's affecting a lot of things, and this can either make Mercury worse or like Mercury can make it better. So like we don't know that yet. So like that's why we're getting this information now, like this baseline information, so that we can compare that like spatially, so to other like food webs that are being studied in the Arctic, and then also over time to see like how it's changing, whether it's going to continue to be a problem, or like if it's going to make those food sources like Arctic char unsafe to eat. Hopefully not, but
Right.
yeah.
And I'm assuming the reason why climate change would affect potentially Mercury in one direction or the other has to do with movement of the organisms to sort of chase down their preferred temperatures, and therefore moving the mercury along with them. Is that sort of how it's related?
Yeah, that could be one aspect. Another is just like with coastal erosion, like there's a lot of mercury that's in sediments, and with like melting of thawing of permafrost or like coastal erosion, that's bringing in a lot of mercury into the environment, or with like food web structure, like whether how those predators and prey change and shift in their roles, that can also change how mercury is going to biomagnify in the food web.
Well, that's the thing with climate change, is that it's hard to predict even how things will change, right? Because it's all unprecedented for our history.
Maybe we'll get the next one right.
There is no redo, dear.
What?
Yeah. Okay, so what does the future for Isabel look like? You're doing your master's now. Do you know what you want to do next? Do you know what you want to do for your career? What are your thoughts right now?
Well, my thoughts right now on like once I finish my master's, I'd like to do environmental consulting for a bit, just to get some more experience. And then hopefully by then I'll be able to go traveling again for a bit. And then I'd like to pursue my PhD.
Amazing.
Very
It does
cool.
sound like you like to travel.
Yes.
Solo traveled over 15 countries. Tell us a little bit about that. Like, was that all in one shot?
No, like different times. Like I did a lot in one shot, but like not all of them.
Okay. What was the-
Yeah. What are your top five
Katie's
solo
top.
travels?
Katie always loves
5
the top,
to 1
top 5.
for
Going
for
5 to number
if you
1.
can think of 5.
I mean, the coolest place I've been is probably like it wasn't solo traveling though, but like the coolest place I've been is probably Ethiopia. That was pretty awesome. Yeah, I'd love to go back. But for solo traveling, I think Turkey was pretty cool. Peru was awesome. I mean, they're all great. Every country has like its own unique flair. So I mean, I don't
Like
want to
what
discriminate.
like I guess what is like one highlight of Ethiopia, one highlight of Peru, one highlight of Turkey?
A highlight of Ethiopia would probably be that I got to spend time in like a very rural area, like pretty remote, and like camped there for a bit. So I got to meet the locals, and so that was really cool because it's like different than just going somewhere and like going to like a city or like even just going to like a national park. It was, you get to actually, I like connecting with locals and like just like learning about what their life is like, and and also learning learning a bit of their language and stuff, because I also really like languages too.
Wow. What about Turkey?
Highlight would probably be, I was at like this hostel where it had like this like rooftop area, so like you could like look over, you could like see like the Blue Mosque behind you, and like look over to like the bay and like just the city. So it was really beautiful. And also that they had like when I was there, they had like a soccer. They're really crazy about soccer, so they had like a soccer finale, and so everybody was out in the street, just like partying, and like, yeah, it was pretty cool.
Did you get a massage?
No, I didn't get a massage.
They have those Turkish massages. They're like famous. They're famous there.
All right.
Yeah, well, no, I'm just saying they look really intense. I'm kind of scared. I kind of want one, but I'm also scared.
I'm scared why that's your first question of someone, a virtual stranger, a guest on our show. You get straight to a very intimate question.
Yeah. Well, I didn't mean it in an intimate way, but I guess that's very
We're all uncomfortable.
Yeah, okay.
This is an uncomfortable moment now.
All right, so who'd you hook up with in Peru? Okay. No? Sorry, too far?
We are cutting the massage section of the podcast.
Sorry, Isabel.
Oh, no worries. Fortunately, but now, like, if that is a thing in Turkey, like now I feel like I missed out. I need to go back now.
You know what's hilarious is I was going to ask you, did you have Turkish coffee? And I'm like, that's getting a bit too proby. And then you come out.
I just went full. I went all the way.
Yeah.
Well, my brother went to Turkey, and that was like one of his big highlights.
Well, that's your brother, for God's sake.
Okay, I'm sorry.
Sorry, Isabel.
Oh, no worries.
So you must really want to be able to travel again. This must be killing you.
There's no traveling.
Luckily, I mean, right now I'm writing my thesis, so I can't really go anywhere anyways.
Right.
So yeah, being stuck at home.
Maybe it's a
yeah, I guess, yeah.
It's hard to call COVID a blessing.
I didn't mean COVID.
Right,
I just meant that she can't travel.
Yeah.
The fact that she can't travel.
The timing is fortuitous.
Hey.
Yeah, let's go with that.
Did you sort of finish all your field work prior to everything being shut down, or did it affect your field work at all?
Luckily, I did. Like, I was supposed to go back up again, but luckily when I was there in 2019, I got a lot of samples. Like, my project is also like, I work a lot with DFO, so Fisheries and Oceans Canada. So luckily, we got a lot of samples. So I was good. Like, I luckily have enough to do my thesis. But I know some people that they had to totally change projects because they really relied on that second field season.
Well, yeah, like you're very constrained in field ecology by the numbers of whatever seasons, I guess, your study species are active. But like if you miss one, that's like a whole year. Yeah. You have to wait for that season again, right?
Yeah, it'll be interesting in ecology to see all like when papers start to get published and you see like that gap.
Yes.
The 2020 gap in the data.
But also how it's going to be affected, right? Like, because human behavior changed so much, even if it goes exactly back to where it was, true. Like all the changes that might have occurred in that time.
Yeah, the absence of human traveling all over the place could have an effect too.
Mm-hmm. Well, it sounds like your project's going great, Isabel. We can't wait to read all the papers about all the cool stuff you come up with.
Yeah. I can't wait to learn more about the mercury I've eaten.
I can't wait to finally understand stable isotopes.
Yeah.
We are all looking forward to that.
Yeah, every time I would think I got it and I try to explain, I'm like, no, God damn it. Well, very cool. Well, Isabel, I think that's it.
I think that's it.
Oh, man, thank you so much for taking the time to be on our little show.
Yeah, we really appreciate getting to chat with you and learning all about this amazing stuff that you're doing.
Thanks so much for having me. I had a great time.
Awesome. Great. Yeah, and hopefully you can get back to traveling soon.
Yes, hopefully.
Thanks, Isabel.
Bye.
Bye.
Oh, wow. That was super cool.
That was very cool. I mean, yeah, there's a lot of mercury in our environment, and we can eat it very easily, I guess. The mining industry, volcanoes, petrol. I mean, we're getting it from every corner, huh?
People smashing thermometers in back alleys for fun.
Well, sometimes people do it by choice.
Yeah, well, that's like the whole thing with the Mad Hatter, right?
Yeah, they used to use mercury in the production of hats. Yeah, and then you get the vapors all day.
The vapors?
I got the vapors. Yeah, but yeah, there's so many chemicals. I mean, I hate to use the word chemicals. Everything's a chemical. I don't like the word chemical as a pejorative, but there's so many toxic chemicals that are floating around in our environment, in the air, in our water, and even in our food. It's hard to avoid, but so it's great that there are people studying this to see what we should be doing about it.
Yeah, and I have a lot of optimism that bacteria will fix a lot of our problems.
I'm going to eat as many as I can.
Not by eating them necessarily.
That's my go-to.
But I know, you can't just eat all your problems.
I think I can.
We need to talk.
All right.
We need to talk more in depth about that concern. But like they're finding bacteria now that they can get to eat plastics and stuff, right?
Amazing.
I know.
Bacteria.
Into what though? Microplastics? That's a whole other
Into
problem.
mercury. It just caused a whole other problem.
Yes.
Bees come out. Giant hornets.
Chemical webs.
Yeah.
Yeah. Everything is connected.
Ugh.
We ruin everything, don't we, humans?
Well, we're just part of it. We're part of the web. Not a great part, but a part.
Not the highlight.
No, we're the mercury.
Oh no.
Yeah, that's us.
We were Mercury the whole time.
The Earth is biomagnifying us.
Oh gosh. Wow. Okay.
Mhm.
I like these connections you're
Yeah.
making.
Sorry, planet.
It's deep and
All right.
depressing.
Well, that's our show.
Yeah, well, we need to go. We've learned today a lot about mercury and food webs. We've
Yep.
learned we need to get some self-confidence. Those are my big
Yeah.
takeaways today.
Yeah, I feel more confident already.
You do?
Yeah, and on your behalf too. I think I feel great about you.
Okay. Are you going to stop eating the thermometers?
We'll see.
Okay. Well, Vinny, can you send everyone out on our socials?
Of course I can. If you're interested in following us on our social media, you can check us out at LRG Pod on Facebook, Twitter, and Instagram. That's LRG Pod.
Don't forget MySpace.
You can forget about MySpace since we're not there.
And what about grad students who are listening to this right now who think that they might be a fitting guest?
If you'd love to be a guest on our show, send an email to learnrealgoodpodcast@gmail.com. That's learnrealgoodpodcast@gmail.com. Sign up today.
Wow, I'm gonna buy that product. I love how you switch into sales mode the second that it's your time to shine at the end.
10% off. Sign up now with code ABC. There's no discount.
ABC?
We don't sell it. It's
Why
a terrible
not
code.
LRG?
Oh, I should have switched it.
Oh well.
We don't actually have a discount code because we're not selling anything.
No, we're not. But if you'd like to sponsor our podcast,
Send us 3 mercury thermometers.
okay, that's it. All right. Thanks again to Isabel, our guest. Thank you to our listeners. Thank you, Vinny.
Thank you, Katie.
Okay. See you, folks.
See ya.